Feeding ecology and trophic role of sea urchins and tropical seagrass community. Background. So some important terms to know are trophic level, which is the position an organism occupies on a food web, grazer, which is an animal that eats grass, biomass, which is the total weight of all the organisms or the target organisms in an area, detritus, which is decomposing plant matter, and epiphytes, which is algae, in this case, that grows on seagrass. So it's important to know for this study the tropical seagrass ecology. So tropical seagrass ecosystems, the primary producers, as you can see here, are seagrass, microalgae, or macroalgae, microalgae, and detritus. And so sea urchins, the target organism of this study, feed mainly on seagrass. And so you can see here that sea urchins and seagrass are connected to many different things in this ecosystem. So if they were to somehow die off or their population were to decrease, it would greatly affect the ecosystem. So the biological question of this study is, how much does sea urchin grazing have an impact on the growth and abundance of seagrass and algae community in Bolinao, Philippines? And so this is important so that we can understand kind of how these things affect the rest of the ecosystem. Their hypothesis was that sea urchins, because of their apparent abundance and feeding habits, were predicted to be the most important direct grazers of seagrasses in Bolinao. So methods. The location of the study was Bolinao, Philippines from October to December 1991, and they did their study at these 5 sites with the dots on the map. So they used many different experiments and observational studies to get their data and come to their conclusions. They looked at seagrass abundance. So they looked at how much seagrass was at each site. They looked at seagrass growth. So they used data that a different organization had taken about the growth of the seagrass lawn, basically. And they used data about the length of the seagrass shoots to calculate the seagrass growth. They looked at the distribution of sea urchins. They just had scuba divers go down and basically count the sea urchins in a given area at each site. And they also looked at the size of the sea urchins. So they took 100 to 150 sea urchins and measured the diameter. They looked at the sea urchin feeding habits in a field study where they collected sea urchins and basically dissected a sample of them and looked at what was in their digestive system under a microscope. Then, based on what they found, the 4 most important food sources, they then took sea urchins into their lab and started them for 2 days, and then you would give the sea urchin one of the types of food, see how fast it ate it, see how much of it ate, and then they would again starve it for 2 days, do the same thing with another type, and they did this with multiple urchins. Then they also looked at the digestion rate by dissecting a sample of sea urchins from each site and looking at what was in their digestive tract.
Okay, so as for the results, the first section will talk about seagrass and macroalgae abundance. So Thalassia hemprichi was the most dominant seagrass species in shoot density and biomass across each of the study sites, with the largest difference in abundance between sites. Other species like Inhalus acorides and Cyanodosea cerulata were also present but not as abundant. Macroalgae, particularly Sargassum crassifolium and Amphoreus fragilisima, were abundant in certain locations and had considerable variation, as shown in the graphic or in the figures to the right. So moving on to urchin density and size distribution, Trimnessus gratilla and Salmaxis spheroides were the most common sea urchin species, and the island Lucero had the highest density of sea urchins overall. The size distribution of the urchin populations also differed between the different sites, with Lucero having smaller urchins overall while having the highest density compared to the other locations, such as Malinup and Silakwi. So the feeding habits of the urchins mostly ate seagrass debris, algae, and living seagrass fawns. Trimnessus gratilla had a higher specialization in diet, preferring T. hemprichi live fawns. Salmacis spheroides showed a more varied diet, including live and dead seagrass fawns, algae, sediment, and among other things. And feeding habits varied between sites overall, with differences in the relative consumption of different food items observed. So certain sea urchins, depending on which site they were at, would eat different food items more than others. The ingestion and grazing rates. The ingestion rates and grazing rates were size dependent, with larger individuals of the urchin population consuming more overall. Total consumption rates varied by species and by sites, with spheroids consuming most seagrass, mostly at the island of Lucero. The seagrass grazing rates per unit area also varied between species and sites, which shows implications for seagrass bed dynamics. Finally, talking about the food absorption efficiency, both sea urchin species studied, which is Trimnessus gratilla and Salmacis spheroids, had similar food absorption efficiency, with Thalassia hemprichi digesting the easiest for both species at 62 to 71% efficiency. Dead seagrass fawns weren't as efficient in digestion for both the urchin species, with having an efficiency of 55 to 64%. And T. gratilla did consume brown macroalgae, while spheroids did, and was able to absorb around 60% of the nutrients. And the red algae, Amphorea fragilisima, was only consumed around 23% efficient from both species.
Okay, so for the interpretation, Tallahassee emercy, a dominant seagrass on the southern coast of Papua New Guinea, is abundant food for urchins and large predators on the reef flat at Bellino. It accounts for 56% of above ground biomass and 64% of real production, with growth rates similar to Philippines and Papua New Guinea. In the ecosystems of Bellino, urchin graze certain plants, including Sarsaparilla, Crasspholium, Hematias, and Amphiorina forgaceum. Although seagrass californium is a common in some urchin environments, urchins do not consume it because of its low amounts of energy, fat, and protein, as well as its low ash count. However, because of its chemical deterrence, herbivores find it repulsive. According to this study, urchins mostly feed on low organic matter, algae, Amphorea flagaceum, and they only take in 23% of the organic matter from the algae as opposed to 55% from seagrass fronds. Although with Trinotis vercosium, urchins also consume 5-ferritin, which makes about 30% of the dry weight of seagrass fronds, according to research. The significant concentration of inorganic materials is 5-ferritin results in low nutritional value. Every day, adult Trinotis vercosium ingests 0.6 to 0.7 grams of DW per Tallahassee testinum urchin. Trimnesius vercosium and other urchins eat older, more epiphytic seagrass leaves. This may be their main food supply. Nevertheless, because 5-ferritin has a poor nutritional value, it also consumed by them and accounts for 30% of seagrass fronds. There appear to be an inverse link between the rate of seagrass consumption by Trinesius gratilla and Saclysis saprophytes, which is related to their body weight. It is mostly carried out in the Caribbean, where large urchin populations graze 50% of Jamaica's seagrass harvest. Seasonal differences in the quantity and structure of urchin colonies affect the seagrass effects of grazing. Population peaks at around 9 urchins between November and January. Urchin counts declined about 0.1% between March and July. Due to urchin grazing, the Bellino Reef fishery loses 24% of its seagrass productivity. The feeding of 5-ferritin accounts for at least 23% of its production. Euphonic contributes 20%, and juvenile fish and herbivore urchins contribute 3%. More than 60% of the 5-ferritin produced in North Shore environments is produced by mesograzers. There must be a quiet trophic flux between living seagrasses, 5-ferritin, and mesograzers. On the reef flat near Bellino, 73% of the seagrasses frond is ungrazed. Food is returned as feces in 30% of cases. Based off the researchers' interpretation, I agree with their interpretation of results based off their research and experiments they did for the study. I agree because there are convincing correlations in results sections such as experiments, availability of seagrass and microlage, urchin abundance and size distribution, feeding habits of urchins, intergestion and grazing rates of urchins, and food absorption efficiencies of urchins, which lead me to believe they interpreted the results correctly. Based off their results, this is a significant discovery because sea urchins are a keystone species which maintain the equilibrium of the ecosystem, and seagrass helps the ecosystem to raise dissolved oxygen levels.
The conclusion. Urchins and other herbivores feed on live fawns of seagrass and dead seagrass debris. Llia hemprichi is the most common seagrass, and this represents about 56% of the seagrass population. As saprophytes absorb organic matter more quickly than D. gratilla, which shows how species have specialization and the outcome of a study can vary drastically based on what species are present. Phyrephydon does not contribute substantial energy to urchins and other herbivores because it's only 10 to 15% organic matter. The rate of consumption by seagrass Llia Isley is proportional to its body weight, and the relationship between weight-specific feeding rate and body size is inverse. Direct grazing is more common in tropical versus temperate climates, and this influences grazing rates in Boliano, where this study was conducted. This is an important finding because seagrass is a vital aspect of the ecosystem and increases dissolved oxygen levels, while sea urchins are a keystone species, which means they keep the ecosystem balanced. This is significant because climate change is negatively impacting the health of marine ecosystems, and certain species are struggling to thrive. If this balance between seagrass and sea urchins is thrown off, there may be harmful ripple effects throughout the entire ecosystem due to sea urchins being a keystone species and seagrass increasing the dissolved oxygen levels, which keeps the habitat healthy and balanced. Future ideas. As of now, very little research has been done on alternate trophic pathways in tropical seagrass communities, meaning that future research should entail looking into different trophic maps and food webs. Similarly, detrial pathways are not currently well understood, and future studies should explore this because it will help researchers gain a better understanding of the role of organisms such as urchins. And just to reconvey, detrial pathways occur when primary producers eventually form litter due to lack of consumption, and decomposers feed off of this material. Similarly, most studies on the role of urchins and other grazers in seagrass beds have been in the Caribbean, and location can severely change the results of a study, such as how in Jamaica sea urchins grazed about half of the seagrass population. This displays the need for more studies in different locations so researchers can find averages to determine a more representative sample of how the seagrass population is impacted by urchins. And this is our source. Thank you.